Connecting.
Perfect. So hello everyone. We are so happy to see you again after this summer. We hope that you are full of energy for this new year. And what the best way to start this year with an EcoMito Club presentation. And so today we have the pleasure to have Emma, a PhD candidate. She is working with Wendy Hook, and today she will present her work on mitochondria, of course, and she will talk about sparrows as well. So Emma, the floor is yours.
Great, thanks so much for that introduction. Yeah, so today I'm going to talk about my first chapter of my dissertation, which is published in Scientific Reports earlier this year. And later on, I'll have a QR code. You're welcome to scan it if you haven't gotten a chance to look at the paper and you're interested in it. But yeah, so the focus of this talk is going to be exploring mitochondrial respiratory function between migratory and non-migratory crown sparrows. And really this work has segued into a lot of focus on the crown sparrows in general for my dissertation, and now for Wendy's future plans continuing this work. So I'm excited to share this with you today. Ooh, make sure. Okay. So the idea behind this work was we had this hypothesis that, oops, skipped a slide on accident. All right, I'm gonna reverse real quick. I'm not sure what happened right there. So first of all, if you are familiar with the migration physiology research and literature out there, great. If not, just a brief overview. There's many adaptations for avian migration that have been identified, and this has really been a focal research point in the past couple of decades of people investigating these physiological adaptations. So we know that avian migrants undergo these biannual movements, predictable movements that require a lot of energy. You have things that cross the Gulf of Mexico over in the Americas, like a ruby-throated hummingbird that has to travel over 500 miles nonstop, or in the extreme example, the bar-tailed godwit that can fly up to 11 days straight. And so the question has always been, how do these birds undergo these adaptations? And so we know that they have whole organism level adaptations where they can increase their metabolic rate, increase aerobic capacity through hemoglobin and hematocrit levels. There's also tissue level adaptations where they have plasticity and organ size, muscle sizes, capillary density have shown to increase. They also have an amazing ability to store lipids and utilize that for fuel. And then there's these cellular adaptations, and that's what we're focused on. And this has been kind of a new avenue that people have been looking at, but still not a lot is known about the subcellular level of adaptations when we go to organelles within the cell, and specifically within the mitochondria. And so that's what we wanted to focus on. So the adaptation is that you have all these different levels of organization with adaptations occurring, so that we would predict that there would also be adaptations occurring within the mitochondria as well, considering it produces over 90% of ATP. And so the prediction would be that migratory birds would have higher maximal respiration rates, mitochondrial density, and higher electron transport system enzymatic complex activities when we compared it to a closely related non-migratory species. And a big question when we started this, was what comparison are we going to use? What migrant and non-migrant are we going to use? And the other thing is, would we see these fixed effects across seasons? Or like all these other adaptations I just talked about, would we see plasticity and flexibility, which we see in these other adaptations going on the physiological level? And so for our study design, we selected 2 subspecies of white-crowned sparrows. This map is a little confusing, but it's just to show you that there's 5 subspecies of white-crowns, currently accepted 5 subspecies of white-crowned sparrows, in North America. And so they're readily available, and that has made them and they're very abundant. And so that has made them an excellent candidate for studying physiological mechanisms. A lot of people will probably be familiar with John Wingfield and Marilyn Romanowski's work on studying endocrinology of gambels and nuttals, white-crowned sparrows. And so we selected these two subspecies because they both co-occur in California, and the gambels is a migrant and the nuttals is a non-migrant. So nuttals are non-migratory resident species along the California coast. They can be found year-round, so they don't move all that much. And the gambels can be found in mid to Southern California over the winter period and pre-migration, so early spring period, and then they breed up into Alaska. And so for this, we wanted to hit certain critical time points. And these critical time points were dictated by the migrant species. So our first data point was in April 2021, where we went out and collected gambels. And where we collected them was this place called UC Davis, where university where John Wingfield and our collaborator Jesse Krause has done a lot of work, and they have shown that this is a well-known or well-established overwintering site for the gambels. And these gambels that winter here will not leave until they're ready to head north. And so it's a wintering site where they will overwinter, and then come spring, when photoperiod changes, they'll start preparing for their migration. We didn't collect the nuttals during this time point because they were already well into breeding. And so we had some concerns about that and what that would look like when comparing, because since they were in a different life cycle stage. For fall mid-migration, we returned and collected both gambels and nuttals. Our nuttal site was always one location. It's called Marin Headlands, and it was part of the National Park Service, and we were able to collect the nuttals there. And the gambels, they occur a little bit more inland in the winter time point during the winter, as I've already mentioned, at UC Davis. And then, oops, I realized I confused winter and fall. Sorry, sorry, y'all. But anyway, so essentially the residents and the migrants sort of split. There's a little bit of an overlapping area, but the gambels are going to be found more inland. And so for fall mid-migration, we wanted a site that we knew that birds did not stop and winter at. This was the key, was where is a site so these birds have a range at which they winter, but where do we know they do not winter, but they will be in active migration? So they are exhibiting migratory restlessness, Zugenru, in that active migration phenotype. And so thankfully, because of all of the work that previous researchers have done, we were able to collect birds and select a site called Tioga Pass. And so this is a higher elevation in Yosemite National Park, or just outside of the park. And we know that the birds that pass through here are not going to winter. They're going to keep passing that site and head further south in California, where they will overwinter. So for the methodology, this was before we had our Ouroboros, so we were using the Hansa-Tech system. And essentially, this is just a schematic of an overview of the process and how we were processing these samples. So we would first go out and collect our white-crowned sparrows. We were focused on the pectoralis muscle, since we know that this is a highly plastic organ, and that selection probably acts on it because it is crucial, really key to flight and flight efficiency. And we excised that muscle, and then we spun it down to isolate our mitochondria and to ultimately get our mitochondrial pellet. And then we were testing this with the Hansa-Tech oxytherm system So we were ultimately putting the isolated mitochondria through the system and measuring oxygen consumption as a proxy for our mitochondrial states. And we use a couple different substrates to do this. And so I'm not going to spend a lot of time on this because I know that you all are mito people. And so just to go over our substrates, we were looking at complex 1, and where we were putting pyruvate, malate, and glutamate as our substrate cocktail. And then we were also looking at palmitocarnitine for the fatty acid component, since we know migrants use fatty acids quite often. And then we were also using succinate as a substrate as well. And so for our terminology, just to define it, since sometimes there is variation, state 3 is synonymous with maximal respiration. And so we're just looking at the maximum rate at which ADP is converted to ATP. And so when we went out to the field, we were using a variety of different trapping mechanisms to catch the birds. And but one of the most prominent or effective ways to catch these sparrows is our potter traps, and using just seed bait piles, in particular when these birds were migrating or during the wintering periods. Sometimes it was quite easy to get them trained to come to these seed bait piles and catch them and process them. And so this is me at UC Davis. You can see the vineyard, the research vineyards behind me, and we were out collecting these birds where the birds were sort of gathered underneath these olive trees where they were foraging. And so we would collect them, and then I would look at morphometrics, collect important information like age based on molt. And then we would go into the Mitomobile, which is our outfitted laboratory that we can use to go out to places like Yosemite, these national parks where there's no lab that we can utilize. And so we bring the lab out to us. So for the results, I chose this one because I think it captures the overall pattern that we were observing within the results. So there are many more figures. And again, if you want to look at this, feel free to take a screenshot or a picture of the QR code if you want to look at this at the paper later. But essentially, just to give you an overview, the orange is going to be the migrant group. The blue is always the non-migrant, so our nuttals. On the x-axis, we have the different time points at which we were collecting, and the y-axis is our respiration rate. And this is looking at state 3 using palmitocarnitine. So first of all, within the residents, you can see that they had this slight uptick in overall mitochondrial respiration in the winter, but it was not significantly different. But really key here to focus on is the pattern occurring within the migrants. So what we see here is in the spring, when we were collecting the migrants before they migrate, but when they were already undergoing these physiological changes to ramp up for migration, we see that this is higher, the spring time point is higher than winter, but it's highest during the fall active migration time point. But then in the winter, when we look at it from a mitochondrial physiological level, these birds are virtually indistinguishable from the residents when we look at them in the winter. And this was the underlying pattern that we observed across many of our time points, or excuse me, many of our substrates. So this seems to be, this was a substrate independent pattern for the most part, where regardless whether or not it was the pyruvate, malate, glutamate, or the succinate, or the palmitocarnitine, we were observing this pattern. Now, what is going on with the residents? Well, thankfully, like I said earlier, many people are studying nuttals and gambles, and the literature has really shown that for the nuttals, their energetically demanding period or a life cycle stage is the winter time point when you have high winds along the Pacific coast and you have colder temperatures and it's rainy. And so we research has shown that this is kind of a an energetically demanding period for them, can be stressful. And so this is probably related to winter thermogenesis, which is also what our citrate synthase data shows, is that they also had a little bit of a upregulation of that in the winter time point. And so overall, what did we find? Going back to our original predictions. And so just to get you acquainted with this graph, this is just to show you a summary of what we found. On the left column is our Gamble's group. The right column is our Nuttall's group. And essentially what we're showing is that when you see a black asterisk, that means that that time point was always higher, regardless of what it was compared to, whether it was compared to the Nuttall's group or within that same, within the Gamble's group with the other time points. And so that fall time point was higher for a mitochondrial respiration state 3 than the Nuttall's time points, regardless of the time point we compared it to. And what we can also see is that citrate synthase was always higher in the migrant group compared to the Nuttall's group. Now for the complexes, and so this is measuring, not to confuse it with, I know there's an Ouroboros measurement to look at ETS, but this is enzymatic activities, just individually looking at the complex enzymatic activities, measuring just with their own assays, individual assays. We really didn't see any patterns that told us that, at least for the individual, the performance of each complex individually, that this seemed to not be the driver of the pattern we were seeing. And while citrate synthase was indeed increased during the spring and the fall, the pattern overall also didn't totally explain what we were seeing with respiration. And so the big question is, of course, now what are the mechanisms that are driving this pattern? And so for future directions and discussion, we did see that, in fact, the migrants did have this upregulation of mitochondrial traits related to migration, and that this plasticity or flexibility was really limited for the non-migrants. And migrants had the highest mitochondrial respiration at peak migration. And so during that very energetically demanding time point where they're actively migrating or exercising, is when this was the highest, and that this could be, evidence that mitochondrial adaptation is essential for migration. And while we found plasticity in both the migrants and non-migrants, the non-migrants, again, based on the literature looking at winter thermogenesis, we believe that that effect is just related to thermogenesis, where the birds are just increasing their number of mitos just to produce more heat in the winter. And our complex activities results was really mixed. We didn't really see any straightforward patterns there, but of course that was just measuring complexes individually. So that's not to say that they're not working in concert, and that's how they're producing that pattern, because obviously they work in concert with one another. But yeah, so for future directions, I've essentially expanded this to other white crown or other crown sparrows in the same genus, and really excited to share that work maybe with you guys in the future. And so with that, I'd like to acknowledge the people who really made this work happen, the field crew, as well as my co-authors, over in kinesiology, and Jeff Hill and Wendy Hood, my co-advisors, and our labs, Hill and Hood labs, and co-advised by Wendy and Jeff, and really just grateful for everyone who helped to make this happen, and also would like to thank our funders. And so with that, I will take any questions.
Thank you very much. I'm trying to react, but here we go. Cool. Thank you, Emma. That's a really cool study. I'm sure there will be a lot of questions. If you want to raise your hand in the chat. And again, sorry for those who joined late because of the link. I think you can see which universities have more security linked to the emails, because I think they received the list emails late. So apologies for that, but it's being recorded. So if you need to watch it again, you'll have it on YouTube soon. Any questions, or should I start away? I have one right away. Oh, Karim, please.
You can go, no worry. Have time.
No. It's because I'm still in EBEC. I'm still with the structuralist. So I was going to ask about supercomplexes. Could that be a mechanistic explanation? Because you see in some species exercise increasing the proportion of assembled supercomplexes, more complex one, more bigger supercomplexes, or different assemblies. Is there any plan to look at that in the future?
Yeah, Wendy is going to be working on that. I am probably going to be finishing up before that really comes into play. Now I'm looking at, for my third chapter, which is what I'm currently working on, we took samples and fixed them in resin from our fall groups. And so I'm doing mitochondrial morphology right now to see how the individual mitochondria structurally, if there's any differences, as well as compare mitochondrial density to our citrate synthase data. But yeah, Wendy is currently working on getting or trying to get the training for that, because that's something she's going to go back with, hopefully with you know, some more students and more funding to look and see, could super complexes possibly be explaining that? Because yeah, obviously we were just measuring individual complexes, and that wouldn't show, you know, super complexes at work.
But I'll note that we did look at mitochondrial dynamics markers, which is going to link nicely to the morphology data that Emma's describing. And I think those results are incredibly exciting. And it's in review right now, so hopefully that'll be out there for you guys to see soon. But it seems that fission and fusion seems to be really important, or at least strongly correlated.
So biogenesis of mitochondria happening as well, or yeah.
We did not measure PG. Oh yeah, we did measure PGC1 alpha. I don't remember those patterns precisely, but the fission and fusion markers, the mitochondria coming together and splitting apart, we know those both increase with exercise, and they're increasing following the same respiratory patterns here.
That was the strongest. Yeah, that was the strongest relationship. And overall, like when you said, that followed the pattern that we were seeing, where, you know, spring and then fall and then down to winter. And so it followed that flexible pattern that we observed. But yeah, there were other things that also produced interesting results on that. But I'll share that paper if you're on ResearchGate, or if you follow me on, I didn't change this, but X, then, you know, I'll be sure to share that once it's published.
Yeah, very cool. Carrie.
Yeah, thank you. Hello, Emma, and thanks for your presentation. Sorry, I cannot switch on my camera somehow. I very like the way you explain your study. It was super clear, and I very like it. So, have you also looked at the state 4 of your mitochondria?
I did, and for the sake of time, I excluded that here. But interestingly, state 4, while it didn't have necessarily as many significant differences, my state 4 data really did follow that pattern, same pattern, where for the residents, there was really no significant difference, although there was a little bit of upregulation during the winter period. And then again, we see that pattern occurring with the migrants. So it was really the same pattern, just some of the in between groups were not significantly different. That was really the only difference there.
You mean the migrant birds, they increase their state four during migration?
Yep.
Yes. Yeah.
Okay. I would have expected.
Yeah. Yeah, I know. So it really follows that pattern. And in fact, that's why we also looked. So if you go to the paper, we also looked at RCR. So divided the state 3 by state 4. And like because of the way that, at least for palmitoyl carnitine, the way that they upregulated state 3 and state 4, it actually looks like, so there's like no difference in RCR.
Okay.
Because both are increasing in the same direction, proportional to one another. And so there's actually no significant differences between the time points for gambles when using palmitoyl carnitine, which was an interesting thing, I thought, when looking at it that way.
And that was an isolated mitochondria, is it?
Yes, isolated mitochondria.
Wow. And do your bird, last question, do they eat during this migration?
Yes.
Do they eat like a lot? Like, I mean,
so they don't eat so for these sparrows, they're not crossing like a huge body of water or anything like that. So the fasting periods, you know, from what we know and from the tracking work, they're probably resting and then flying for a few 100 miles and then resting because they can, you know, they have that option. And so they do opportunistically forage. And then of course that pre-migration time point, we know that they undergo hyperphagy because they have to somehow have increase their energetic output for hypertrophy to be occurring, like to increase their protein stores, to increase their fat, adipose tissue stores. And so they are eating way more than they usually do during that spring time point.
Okay, well, thank you. And I would be very curious to know whether they actually produce more ATP, but I suppose that would be a full job. Thank you.
Thank you.
A quick follow-up on that, because I was going to ask you that, but I see there's a couple of questions. But I remember when my first undergrad project was with Jean-Michel Weber in Ottawa, and his lab had just shown that these migratory sandpipers were gorging on omega-3 fatty acids before migration. So when you talked about the not all ones that were on the coast, I thought, oh, maybe they have access to good migratory food. Because they were eating those little shrimps, the sandpipers, and gorging on DHA. And I was going to ask you, do you think that's happening as well with the migratory ones, that they're, and so that's why the palmitoyl carnitine is so high compared to the non-migratory ones?
Well,
but you basically answered,
yeah. So essentially, because the migrants are more inland. It's the residents that are on the coast. But neither are going to be eating like crustaceans or anything like that. Because so typically songbirds, they do increase, the literature has shown that they increase their fatty acid consumption, but it would be totally different types of food. There is studies on that, essentially what they're looking at what they eat during migration. I don't remember off the top of my head how they switched those food types. I would have to look it up. But they do have a shift of food resources. But of note, it was a really, really dry year, the year we collected the birds. And so they were actually, I think the birds were having a harder time finding food resources just because there wasn't much available at the time.
Yeah. Well, I just found a follow-up study on those sandpipers from Chris Guglielmo's lab. And the title says that the EPA and DHA don't affect muscle oxidative capacity. So I guess they can do without. And that's what your birds show as well, right? A lot of plasticity.
Yeah, it's hard to know, right? Because right, because we take them out of the cellular context, plus we don't have that interaction of diet. because we don't know, like we didn't look at, exactly what they were eating at the time we collected them. So I think there's a lot of room to do more to say, because yeah, Chris's paper, I'm very familiar with that one. But there's another paper that shows if they don't have this specific fatty acid in the biofilm, that their flight efficiency decreases by like 40%. So it definitely, I think, could be variable depending on the group, because we're talking about like very different groups, like the sandpipers versus the songbirds. So I think it probably is different, varies depending on what we're talking about, like songbirds, sandpipers, or even genus, like genus and family, bird families.
Cool. Neil?
Okay, so thanks very much, Emma, for a really nice presentation. Firstly, before I come to my question, did you say that UC Davis has a research vineyard?
Yes, they do. Yes.
Oh,
it's
I
like
think my university needs a research vineyard as well.
Yeah, like it's like the only one I know of, and it's a pretty prominent one in the United States. So that's what
everyone
collecting.
Everyone needs to sort out the next sabbatical then, no?
Yeah.
Yeah, very nice.
Unfortunately, yeah, there was no wine involved with that.
Oh.
So.
You would think they'd bring us a couple of bottles, but they didn't.
Okay, on to my question. I was just wondering why presumably the migratory subspecies, it must somewhere be paying a cost for having more efficient mitochondria. Otherwise, yeah, why doesn't everyone have it? So do you know of any context in which this greater mitochondrial activity, greater mitochondrial density and everything is paid? At what point is a cost being paid? Any idea?
Well, I think that this is the difficulty, because I'm really into understanding, like I'm interested in trade-offs as well, and looking at like how different life cycle stages interact with other life cycle stages. And the thing is like, especially with mitochondrial traits, this is such a new approach or new avenue that we don't have a lot of that information in terms of like, how does that impact future reproductive outputs? And how do things like migration distance impact your reproductive output? And so out of Chris Guglielmo's lab, Soren Colson, he just graduated with his PhD out of Chris's lab. He also looked at very similar things to me, but in a different group of birds, and induced photoperiod in the lab. And he was measuring ROS, reactive oxygen species. And he shows that essentially ROS did not increase with increasing efficiency, which I think sort of is counterintuitive, because we would think maybe it would. So in terms of like what cost, I don't really know. I mean, obviously it's flexible. That's the first question. Was it fixed or is it flexible? Well, it's flexible, so they're not maintaining these high rates, especially like with state 4. It's like I would suspect that there would be this negative cost to maintain high maximal and high basal throughout the year. So they're not doing that. But what costs that they could be paying for or trading off with this, I think is something that I'm really interested in pursuing, but I don't really have an answer at the moment. Thanks, Neil.
Hichem, do you want to ask the next?
Yeah. Thank you, Emma. I just have a small question. I'm curious about complex 1 substrates. You also added glutamate. Do birds in general rely on this substrate, or is it this species specifically? Because insects, depending on the species, you rely or not on this substrate. So I was just curious about that.
Well, this is something that actually Wendy and I have discussed quite a lot, because a lot of these substrate cocktails have been just predetermined based on and adopted based on the exercise and like biomedical or like kinesiology literature and studies based on like mammals or humans. And I'm sure a lot of people are familiar with the Kuzmiak 2011 paper where they looked at substrate usage in mice versus birds, and they showed that, you know, birds had like a 7 to 10 times fold increase in capacity when they use like palmitoylcarnitine, or they could more readily use palmitoylcarnitine than compared to the mammals. But I believe the pyruvate malate glutamate was relatively they had the same capacity, the mammal and the bird. And then I can't remember, I don't think they looked at succinate. And so I think that this is also something that is missing, because and this is why I come from an ecological perspective, because it would be interesting to identify like diet, I think, and then say like, okay, how is this related to this? Because a lot of it hasn't been looked at. Like we know that they can use it if we give them the glutamate in that pyruvate glutamate malate substrate cocktail, but whether or not that's actually occurring in the wild and to what degree, I don't think we know. So I think that's also an excellent question.
Yeah, even for insects, when they said that depending on the species, it's actually when you do in the lab, you can check. In the wild, we don't know. So, and also about complex 1, how flexible was complex 1? You said that all complexes were about the same depending on the season. So even for complex 1?
Complex 1 had no changes at all.
Okay.
So yeah, so there were a few complexes that did have some increases or decreases, but a lot of times they weren't significantly different. I would have to pull up my grasp, but yeah, for complex 1, there were no patterns whatsoever.
No, I was expecting that. Okay, thank you, Emma.
You were.
Yeah, because I mean, I was expecting your answer because I was not sure, but I wanted to ask because in insects, I did some comparison between 3 species of insects for different complexes of the ETS, and complex 1 in general is the least flexible. So I was curious about the birds also. So yeah, thank you.
That's awesome. Thank you. Jessica, is it next?
Yeah, Jessica.
Yeah, can we hear me?
Yeah.
Hello, Emma. Thank you for your presentation, and well done for your work, especially with a large number of birds sampled. I know it's a big job. And so the results of the activity of the complexes don't show any flagrant differences. It's all right. But I know that in Scott's studies, particularly on bar-headed geese, the capacity of complex 4 has decreased, but the affinity of the cytochrome C and oxygen has increased, and which means that oxidative phosphorylation is very efficient, even in extreme conditions. So my question is, have you thought about looking at the differences in the subunits of complex 4 between migratory and non-migratory birds?
Yes, I think that for future things that would be really interesting, because like in terms of our capacity with what we have now, we haven't explored like the different subunits, because the question was, you know, is this even occurring since it was so novel, and what are the patterns, so then we could identify specific mechanisms. But yeah, you're absolutely right. In that case, you know, I think too, this is the difficulty, is looking at the phylogeny of the of birds. It seems like, you know, so we can't agree on how evolution has evolved or, you know, been lost and gained throughout these lineages. And like with the bar-headed goose, the question is, would something like a songbird, you know, in my mind, would convergent evolution occur like for latitudinal migration versus something extreme as bar-headed geese, which is flying over the Himalayas? you know, in such low oxygen environments, you know, these birds, these songbirds have shown to fly, you know, upwards of at a few, maybe 1,000 feet. But whether or not that translates into a similar adaptation, I think is interesting. And that's what we're trying to figure out, because this isn't just one group, right? And there's many groups of birds that have very quickly become resident or migratory. And then, like you said, there's all of Scott's work with the extreme altitudinal migrants. So yeah, thanks for bringing that up. I think that is something we need to look at.
Yeah, especially if you are a frozen sample, you can perform PCR and look at for subunits. Yeah,
that's really interesting. Thank you.
Thank you.
But also your hematocrit, the birds, the migrant ones, your hematocrit levels were increasing. It seems to me from reading, looking at all the figures together, that there might be mitochondrial biogenesis, higher complex 1 capacity slightly maybe, but like from spring to fall, they seem to upregulate everything that allows for a more efficient function.
Yeah, right. And so like our citrate synthase did increase a little bit during that fall time point. And then of course, you know, I think that there's a couple things going on, is that I think that personally, I don't think there's one mechanism. I think that, you know, everything can come together to have that increased capacity, but we're obviously missing still the major mechanism. And so I do think that increased citrate synthase does play a role, even though that's not even a perfect measure of number of mitochondria. Yeah, it's like you said, like everything is increasing, and I agree with that everything is sort of supporting that increased respiration, and it's just a matter of finding the major mechanism, you know, to show what is driving that pattern.
I didn't see I didn't read in detail your methods, but in your Clark type electrode, was it a sequence, complex 1 first and then palmitoyl carnitine, or did you subdivide?
They're
the steps?
Separated. So with a hand,
they're separated. Yeah.
You just use like one substrate cocktail per run per chamber. So you have multiple chambers running at the same time.
Right. Any other questions?
Yeah, I think that Neil has one.
Yeah. No, thanks for the
maybe the last one, because Emma answered to a lot of amazing and interesting questions, but maybe maybe it could be the last one.
Yeah, for sure. I mean, and I don't even know if there's really necessarily an answer to it, so don't worry about it too much. So yeah, so I love that you included a bunch of the enzyme data along with your respiration measurements, but they didn't really line up that well.
No.
Right? So the complex 1, I was kind of looking at it thinking like, wow, they don't match up. What do you think's controlling? So let's say if it's completely true and like your enzyme data is truly representative of what's going on, that there's not more complex 1 present in the migrants, what do you think's happening instead?
So Wendy is convinced and has made me convinced that there definitely could be something going on with super complexes. And even apart from super complexes, I think, you know, when I was reading up trying to understand the patterns I was observing, you know, I reminded myself that and they have demonstrated this multiple times, that when they work in concert with one another, like the actual complexes, they can show a totally different pattern. So like I found, you know, an option to do the assay, but like with 1 and 3 together, or 1 and 3, or 2 and 3 together. And so I think that could very well produce a totally different product once you look at its capacity together. And then going even further, looking at, could there be, you know, like super complexes going on? I'm particularly interested in the morphology, and that's why, you know, I'm looking at this for chapter 3, is that I do think that, you know, things like increased cristae density and things of that nature could be a major contributor to this as well, like contributing to the individual morphology that is then contributing to that overall increased respiration. And like Wendy was talking about our biogenesis data, I think that supports that. I think it supports that there are things going on within the actual mitochondria, fission, fusion, that on the individual level, their morphology is changing.
Okay, so I got it right. It's sort of, if the proximity of any given complex is too far away, you're going to see a disconnect between it. Is sort of what you're suggesting there?
Yes, and so what I don't want to, I do not want to sound like an expert on this.
No, it's okay.
Getting into like so from what I have read, yes, like things like proximity to each other. So like if you increase the cristae, not only do you have more room for more electron transport complexes, they also are closer in proximity and can form the super complexes. And so personally, I think that what's going to show is that all of these things like super complexes, when, you know, Wendy gets to looking at this within this system and morphology, you know, again, that's my prediction, is that there's going to be these increased morphology metrics on the individual level, the mitochondria, that's all helping to produce increased capacity.
Cool.
Thanks, Neil.
Thank you. That was a good question in the end, Neil. Not different from everyone else. So I think we should wrap up here. What do you think, Elisa? Unless anyone has a last question.
That's a good idea. Emma did an amazing job in answering all the questions. Congrats, Emma.
Thank you. Thank you very much, Emma, for the talk and for the discussion. I'm going to stop the recording.